Hello.
Hello.
How are you?
Oh, I'm fine. I've managed to figure out the paper I was working with,
Okay.
and I was getting wrong graphs.
Yeah.
I've decided the paper was not. I wasn't wrong. The paper was wrong. There you go.
Oh yeah. What brought you to that conclusion?
Part of the labeling on the graphs for one part of it, and another was that I got the right answers when I accidentally inverted my graph.
Oh.
Really bad.
Yeah.
Anyway, yeah.
Yeah. Adventures in graphs.
Yes. Well, I took the graph. It was like this, or rather, I guess it was like this, curved like this,
Yeah.
and I did this with it,
Oh, okay.
and this was wrong. They drew it like this. This is 90 degrees flipped.
Yeah.
It
Yeah.
looks easy to do, and these simulations. Anyway,
Yeah.
mystery solved. Wasted 2 weeks.
Oh yeah.
Oh
Well,
well.
yeah. Hello, Sushmanth. How are you?
Fine, Reddy. And this week I worked on a lot. I was also stuck like Susan on 2 weeks on the same problem. I couldn't it solve, but today morning I was able to clear it out with the SAM model. Somehow, yeah, my told me to drop the SAM idea, but I was still continuing on it. I'm seeing pretty good changes in the model,
Oh, good.
and my model will be trained. I started working on the DevoWorm model also. Uh, since last week, I was able to give most of my time to GSoC and this. Can I share my screen and show it to you,
Yeah.
my
Go
work?
ahead.
Yeah.
Okay. There we go. I can see your screen. But I can't hear you, Sushmanth. You're muted.
Sorry.
There we go.
I'm audible right now? Yeah.
Yes.
Yeah.
Yes.
After trying out the SAM model in a classical way, I couldn't implement because the model architecture was completely different. Then I found some tutorial on the internet, MedSAM, this repo. They are trying to segment a 3D, fine tune a 3D data set using SAM model. And the major problems using this code repo is I need to change a lot of things because here they are extracting only single bounding box, not more than one. But right now we have the images of this type. Yeah, yeah. Could you see this thing?
Yes.
We need to create bounding box for each cell, and then we need to give as a prompt to the SAM model. But here, whatever they wrote, it is just creating only one bounding box. So I need to change to take multiple bounding boxes. Then I need to start training the model, and I was stuck whole week on the training of SAM model because here there are so many unknown things they haven't mentioned. The code was not pretty clear, and it took me a whole week to understand it properly. And actually, there are three main structures in the segment anything model that are just a minute. If you see this picture, if you give an image, first it will go to image encoder. In that image encoder, it will go and give out some image embeddings which are of different sizes 256, comma 64, comma 64 size. And here we need to give another input also, bounding box points and text. Here we are giving bounding box, and whatever image encoder gives us the output, that will go to this mask decoder. And till these days I was thinking just giving an image, I'm giving directly image to mask decoder. I'm giving mask this image directly to decoder, and it is not working. When I was going through the MedSAM code, then I understood first they are giving to the image encoder, then they are extracting some values. See here, image embeddings. They are extracting some image embeddings, and then they are transferring to the decoder part, and then they are giving the prompts of boxes. I understood and I started. I wrote the code of it. I pushed my whole work into the GitHub. It's a few minutes back. Yeah. Yeah, this is the thing I have been working on. Right now we have the images, 2D, 3D images that we need to convert into a NII file form. So I converted them. I wrote this code and I tried to convert them, and they are converting perfectly. After that, I was resizing the image into this form of size 1024, 1024. Only that size of image can only be taken by SAM model. Not other than that, it couldn't take any other model. So we are passing to the image encoder. This input image is passing to image encoder. Then we are extracting these image embeddings. Then these image embeddings we are giving to the I should have made a PPT. Sorry.
It's okay.
Well, when we give this to this image embeddings to here, it will work properly. So there are little errors going on right now. Maybe by tomorrow it will work. And I started working on the DevoNet model also. Here, segment of Samsung. Okay, just a minute. Yeah, here comes the DevoNet model. Here I started writing the code. First, I wrote about dataset. This part is about completely of taking a TIFF file, because whatever we have the dataset from cell tracking challenge dataset, we have three things here. First one, these are the original images of cells in a TIFF file form, and these are the ground truths of the cells. These are the ground truths of the cells. They are actually blank, but this is how they look when you open them. And this is the original image. And these are like, and in this folder we have, here we have the some dots kind of structure where we can give to the model. So right now I have wrote the datasets file here. So first we are taking a TIFF file as input. Then we have the 3D dataset. Whatever dataset we are using, it is a 3D dataset. So I created a function about it, taking an input a 3D dataset, and I was pre-processing them, rotating and etc. stuff. And I'm taking get image function answer and label function get label. Label means ground truth of it, and trying to pre-process them. This took me a while because I need to understand what is the resolution they took in the microscopy. I need to go through the whole dataset documentation to understand what dataset microscopy resolution they took. According to that, I need to change the parameters here. So I wrote a code of this, and I'm writing this in the PyTorch. Actually, there is a resource in the over internet for this thing, but they have wrote it in the QC. Yeah, no, I think I don't have that file, but they have wrote this code in another framework called Chainer. Chainer is also another framework like PyTorch, but I'm trying to write it in PyTorch and implement it. So the datasets file is over. By next week, I am trying to implement the model architecture like NSN, Nucleus Segmentation Network, Nucleus Detection Network, and a trainer file for that. Yeah, those things I will implement for this 3D dataset. Bradley, do I need to add for 2D also, or just should I go with only 3D thing? Because whatever dataset we are using, it is 3D dataset. But in future, someone wants to use for 2D dataset. Should I add that, or first I will work on this? Or
yeah, I would do that just to have the option to analyze specific frames.
For 2D.
Yeah.
Yeah, like I will create a function for 2D dataset. I will just keep that. If someone wants to use some 2D dataset on different cells, they can directly use it.
Right. Yeah.
Is it okay? Yeah. And these are my updates. Mostly this week I worked on this file, writing this file, understanding, converting it into an image embedding, and the trainer and the data set. Most of the time was went in understanding the SAM model. Actually, the model new, and they haven't. The Meta was so secretive about this model, and they are not releasing much things about it. So fine tuning it is like bit hard, but I'm almost there. I will complete by this weekend.
Oh,
And
great.
yeah,
Yeah.
yeah, and I will start working on the DevoNet also. Uh, there are so many errors coming up, but right now the situation is okay. I have given my update to Mayuk actually. Few minutes back, he texted me up and he was closely watching me whether I'm working or not. Yeah, that's it.
Okay. That sounds great. So yeah, this is of course SAM, which is the segment anything model, and you're trying to sort of build a specialized version for
C elegans.
microscopy.
Yeah,
Yeah,
and
yeah.
yeah, my final goal would be I will be comparing DevoNet model and SAM model, and which model, because SAM is trained on billions of images and DevoNet is just trained on the C elegans. I'll see which is performing better over the cell system, mainly in C elegans. And my main motive would be like making model more perfect with DevoNet model more perfect using SAM and comparing both of them. Yeah, implementing cross section of these two.
All right. Yeah. How much work has been done on SAM, like outside of, again, they have an initial publication. Are people using it for things already, or is this
Yeah,
too
actually
early?
they are. Yeah, actually they are using. Here you could see maybe this is another repository MedSAM. It is mainly used for. I will keep in the channel. Yeah,
Okay.
let it.
Oh, so
This
this
thing.
is, yeah. All right, yeah, MedSam, and that is,
Here they are fine tuning the SAM model for medical image analysis, mainly for lung segmentation, etc. But this can be used for cell level also because it takes lot of parameters. The only disadvantage we have is SAM model, original SAM model was trained on the color images, RGB, 3 channel images. And whatever the cell images we have, it is a grayscale images. So we need to convert them from grayscale to RGB. Then after converting into RGB, we are converting it into some image embedding. So this architecture, someone wants to implement SAM model, they need to get clear idea on the image encoder, prompt encoder, decoder structure. They don't get idea on this thing, they could never implement this model.
huh,
Yeah.
yeah.
Actually, the SAM model was like some big revolution in computer vision right now. It's like a chat GPT for computer vision products.
Oh, yeah. 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 I thought that was what, like, Stable Diffusion and Midjourney were, but
Yeah,
I guess those are
yeah,
more generative models. Like,
yeah.
okay.
We will just segment the objects. We can take out whatever object we want, and yeah, generating images. It can be used in our lineage. Sorry, embryo GAN model is there, right, in DevoWorm. There
Yeah.
also we can use it. It has different type of implementations, but the model was not completely released. They just released the weights of the model, and they are really secretive about this model because as you see, chat gpt is there, the model architecture was not open, same like this thing is happening with this model also. So yeah, this is just a bit kind of problem I was having to implement it, but yeah, somehow maybe by this weekend it will be completed maybe. Yeah.
Okay. Yeah, that's great.
De- DevoNet model is also working fine. By next week, I will try to implement all model architecture according to the image resolutions we have. After that, I will try to implement a loss function for that. And I need to talk with Mayuk once for those loss functions because I don't know which loss function can be used for microscopy images to take it. Some are mentioning it over internet. It is told like dice loss function will be used, and some are telling BC dice loss something another function. So I'm just a bit confused with which one to use. I will talk with Mayuk and I will clear my doubts and I will implement.
Yeah. So the image resolutions are like a lot of these models are trained on image resolutions of a certain size, or I mean, are they scalable, or how does?
These images have different type of noise in them actually. To remove those noises, we need to know about the which type of microscopy they used and which type of lens they used to remove the noise and add some what we call Gaussian noise to it and remove the noises. So yeah, actually, whatever the data set we have, it has pretty clear documentation about microscopy images, like which microscope they have used, and each file represent what, and yeah, different kinds of things are mentioned. But I need only the image resolution they have used and the what each file represents. I have clear idea on the data set what each file represents. In that data set we have three things. One was the original microscopy image, which was uh, took for 30 seconds. Each TIFF file contains of a 30 seconds of C elegans, and for that correspondingly we have dots of each cell represents as a centroid. And we have another thing also, segmentation maps, ground truth of the segmentation. And these all things I need. I thought I need to make my own data set for training, but somehow these data sets are available over internet. I can use them. And the data set is not small. We have 190 of TIFF file images. It is pretty okay to use and we'll get a good accuracy. I'm using 80-20 split for training, 80% for training, 20% for the testing the model, whether it is working or fine. So yeah.
All right.
Yeah. Wait, I will show this thing.
All right.
In dataset what we have actually, just the small thing. Yeah. Bradley, these are the microscopy images, original microscopy image. This is the segmentation maps. And these, could you see these dots? These represent the centroid position of this cell. If this cell is there, for example, the
Yeah.
centroid, this dot represents centroid of that cell in the Z plane. And by this, we can accurately extract the centroid of each cell at different time lapse, and we can segment it. We can extract, as it is a 3D data set, we can extract the volume of each cell also. And future work around this model would be implementing the lineage population model for C. elegans on 3D dataset using this dataset. And main future work would be around lineage population model only. First, we will try to implement these models in instance segmentation. After that, we will try to implement lineage population, tracking the cells, which is the parent cell, which is the daughter cell, according to the colors. Yeah.
All right. Yeah. Yeah, that would be great.
Those are my updates. For next week, complete week, I will work on the DevoNet and SAM parallelly. And I have took permission from my college. They actually provided me with holidays to work on GSoC. So I will give my full time on GSoC itself.
Okay.
And yeah, that's it. My updates are. If anything is there, I will approach you or my. I will think I will have a meeting with him within 2 days for that loss functions, and I will let you know if any improvements are there or if I found out any problems.
Yeah, that sounds great.
Yeah.
Thank you. Yeah, great. So it looks like Sushmanth is making progress on the SAM model and getting that sort of integrated into the DevoLearn pantheon, I guess. It'll be nice to have like this whole sort of, having the three sort of toolboxes or models, and then, having a bunch of stuff that people can use around that. So, not just being limited to one type of approach. And then, at some point we'd like to go beyond C. elegans with this. But we have to start with C. elegans because it's a fairly easy target. So thanks once again.
Bradley, do we have any label kind of thing to label cells actually? Any kind of tool we have to label the cells, which is the parent? Or actually we have we generally don't make data sets in DevoWorm or where. But if we have some kind of tool to label cells, this is a cell of parent cell, or this is a daughter cell, it can be useful, right, for DevoWorm, some kind of a tool.
Yeah, I don't know if there's a
By next
tool
week, I
per
will give you
se.
a small.
Yeah. Go
I will
ahead.
give you a small demo on that. By next week, I have a thing in my mind.
Okay.
First, because right now for me it took around 1.5 weeks to get data set we want to use. I need to understand it. But if we have something kind of stuff, if Susan was taking some microscopy images and sending it to you, we don't know how to make the labels of it.
Right.
What is the parent cell or daughter cell? If we have something which is hosted over the internet, like DevoWorm models in the Heruko or Spaces, is there, right? DevoWorm models.
Yeah.
If something is there on the internet where we can make labels of each cell, this is the parent cell, this is the daughter cell,
Right.
labeling it by AB, over some time it can be used as a data set in future purposes. I can't explain clearly in like this, but I will make a PPT next week and I will show it to you what was there in my mind. And I found out some resources. It just takes around 3 days to implement it over internet and host it in like a web thing. By next week I will show it to you a small demo. If it's okay, I will host it as a DevoWorm tool.
Yeah, that would be great. Yeah, I don't know of anything like that too, so that would be great to do. I mean, people have done this before with like labeling, but it's usually like a biological label. So they're using GFP and they're putting it into the parent cell, the daughter cell, and they can track cells like that. Or sometimes it's like they'll use
ImageJ is there to label these kind of things. But if we have something, we have the SAM weights right now, SAM model. If we use that model, segment it. After that, after segmenting the cells, we can label it, right. This is the parent cell. This is the daughter cell. I couldn't explain this way, but next week I'll show you a PPT. You will get a clear idea what was there in my mind.
Okay, that sounds good. Yeah, that should be quite useful. So thank you for that update, Sushmanth. Look forward to next week. Himanshu messaged me and said he couldn't make it today, so no updates from him right now. Although he'll be updating us in the Slack. I think everything's okay with him. And I don't know if... I don't think Jia Hong will be here this week, but last week he gave a nice discussion about some of his ideas for sort of how to integrate the graph neural network approach with some of the embryo network work that the group has been doing in previous years, and then also some of the more general approaches that one could use for this. So that was a nice presentation. I refer you back to last week's meeting for that. Hello, Morgan. How are you? I don't know if you're available to speak right now, but if you want to give an update, you can put it in the chat. I saw Jesse here earlier as well. I don't know if he's going to be back. In any case, yeah. All right. So actually, I have a thing to talk about that Morgan he brought this to my attention at one point, and last week we talked about creating human embryoids. So last week we had I did a presentation of 2 papers that have been written on human embryoids. This is where they're taking human embryos and they're taking stem cells from humans and they're creating these human embryoids, so they have tissues and sort of the structure of an early embryo. And they're using next-gen sequencing to find, to identify cells by their markers. So when they say distinct tissue, they mean that cells that express certain transcription factors or express genes that are consistent with the things that you find in biology. So, for example, when we talk about a certain type of cell, like a muscle cell, that cell can have a bunch of genes that are upregulated or expressed that match like a muscle cell in an organism. If we just did an assay of your muscle, we would find these same genes. And so, it's said to be like a muscle cell, or sometimes they'll use the term muscle-like cell because, we don't really know. You can do histology on the emerging cells or some of the tissues, and you can see commonalities in the cell morphology. You can do these type of molecular analyses. And then, of course, we talked about the role of implantation in primate embryos, or actually mammalian embryos. If you basically have to have the embryo has to implant in the wall of the uterus in order for it to be a viable embryo, and then make it to give a live birth. So that step has to be in place. Now these embryoids aren't. I don't know if they're implantable. I think they're just growing in a culture, and they're differentiating, and they're forming these tissues. So they call it an embryoid, and this is something that goes beyond some of the organoid work that we've talked about, where you have these three-dimensional cell cultures, and you have different cell types that are derived from stem cells, and you get maybe working in concert, so you maybe form like a system of different tissues. Sometimes it's just an assortment of different tissues in the same culture. And when I say tissue, I mean like tissues that are made all up of these mature-like cells, so like muscle-like cells, or fibroblast-like cells, or whatever, epithelial-like cells. So we don't know exactly if they're exactly the same type of cells you'd see in the biological context, like in the organismal context. We really don't know if they work together in the same way as you would see in an organismal context. But it's promising work. And so this week, this is all a preface to what I'm going to talk about now, which is that we talked about humans last week. Now they also have work in monkeys, so we're going to talk about that. Okay. So this is an article in Nature. This is, of course, was shared by Morgan. Lab-grown monkey embryos reveal in 3D how organs begin. At 25 days old, specimens could be the oldest primate embryos ever grown outside the womb. And so this is the 25-day-old monkey embryo stained with a conventional dye, which is the pink image. So this is the conventional dye, and then this is the fluorescent dye coming up. So you can see that there are multiple colors here. The blue patches in the fluorescent dye denote cell nuclei, so you have a lot of nuclei there. The dense central patch of green and red mark trophoblasts. So you can see the cells as blue, and then these trophoblasts as green and red. Let me do it again. Okay, so green and red are in here, and those are the trophoblast cells. So this is where these are cells in the embryo's outer layer. So this is a 25-day-old embryo. This is what they look like in utero at 25 days. So there's a structure of different types of cells already. You have an inner cell mass, an outer layer, and so forth. We have this sort of structure coming up. And scientists have cultivated monkey embryos in the laboratory long enough to watch the beginning of organ formation and the development of the nervous system. So these are two milestones that are a bit difficult to observe in embryos growing in the uterus. So they're taking these embryoids and they're growing them up to this point where they can observe the beginning of organ formation and then the development of a nervous system. The embryos reach the age of 25 days, making them what might be the oldest primate embryos to be grown outside the womb. So this again is where they're actually growing primate embryos. And so this is actually based on two independent papers from, I think this is from 2023. Yeah, this is from this year. This is 11th of May on 2023. So there are two papers here. I think this Gong paper and the Zhai paper, if I'm not mistaken. Yeah, those are 1 and 2. So the Gong paper, if we look at that, is ex utero monkey embryogenesis from blastocyst to early organogenesis. And so this is by a Chinese group. And what they've done here is they've have a 3D culture system that supports cynomolgulus monkey embryo development until 25 days of development, post-fertilization development. Cultured monkey embryos initiate early organogenesis and recapitulate key events. So they're actually like building a 3D culture system that can support the growth of these embryos, not just an embryo analog. Neural induction, yolk sac hematopoiesis, and early germ cell development are seen. So you see these other milestones of development where you would see this in the embryo. scRNA-seq, which is this next-gen sequencing technique, which was used in the human papers, shows the developmental trajectory of early monkey organogenesis. So this is where they show this trajectory from stem cells to these differentiated cells. So that's this group. And then the second group is Zhai. And I assume this is also a Chinese group. And this is, So this is neurulation of the cynomolgulus monkey's embryo achieved from 3D blastocyst culture. So this is a blastocyst culture. This is a bit different. Cynomolgulus cyberblasts are cultured to 25 days post-fertilization within an optimized 3D system. Cultured embryos recapitulate developmental features of their in vivo counterparts. Early neurulation features are demonstrated in cultured embryos, and epigenetic features of 3 germ layers in primate are unveiled in cultured embryos. So they're actually able to get the 3 germ layer sort of epigenetic features of that. And I don't know what they mean by epigenetic features, but the graphical abstract is here where they have this prolonged embryo culture in this special system. Here you get, it's pretty small, you can't really see it, but you're starting to get neurulation here. You're starting to get a neural plate border and differentiation of the neural crest. Then you get the neural tube forming a little bit later, and then you get the amnion and the mesoderm and the endoderm. So you get all these different things happening in this embryo model. So those are those 2 papers that were published. Let me go back up to the top here, or where we were before. So it's very impressive. This is actually one of the authors of the human papers we talked about, a developmental biologist at Caltech. It's going to bring a lot of new insights. So basically they're going 3D. They're building this 3-dimensional cell culture. So previously both teams had managed to culture monkey blastocysts in petri dishes for up to 20 days. A blastocyst is a ball of dividing cells. So it's like this mass of cells in a sphere. That's what you have a blastocyst. So that's kind of what they had been able to manage previously. And those last 2 papers, 3 and 4, were examples of that work. Past that point, all of the embryos had collapsed, making it impossible to see more advanced stages of their development, such as early signs of the nervous system and organ formation. So in previous work, they've been able to get these sorts of balls of cells, and they were dividing, but they couldn't get any sort of structure. They couldn't get a lot of differentiating cells and keep them in this anatomical context. So they say the embryos had collapsed. That just means that they didn't survive to that stage. So that was the problem that they had. They couldn't get past that stage of development. But in the new studies, the researchers grew monkey embryos in small vials of culture medium, which allowed the embryos to grow in 3 dimensions as they would inside the womb. So they had these 3-dimensional cultures that we saw an example of in the second paper. Both teams coaxed their embryos to survive 25 days after fertilization. And then this is where they have some content here if you're interested about some of this. And so these are, so they obtained egg cells and fertilized them in the lab with sperm collected from their male counterparts. A week later, they placed the resulting blastocysts into a gel-like substance in small cylindrical containers and watched them grow for 25 days. So about 2 weeks after fertilization, more than half of the embryos had an embryonic disc, or a flat mass of cells. These discs eventually form the three main layers of the cell body. These are the three germ layers. The lab-grown embryos also showed genetic features similar to those seen in natural monkey embryos within the same time frame. By day 20, the embryos had developed a neural plate, one of the early hallmarks of the nervous system. As in natural embryos, this plate thickened and bent into a tube that forms the basis of the brain and spine. So this is where they were showing those examples of the neural plate and the neural crest. That's all observable in this model. And you can also see the three germ layers, and then you have earlier stages as well. They also pinpointed cells would eventually become motor neurons. The insights gleaned from the lab-grown embryos will help researchers develop a better understanding of early embryo development in primates. So this is another example in primates of monkeys, not in humans, but you have these two competing. So basically, primates were beginning to be able to do this in different species. So, every species is different. We have model organisms like mouse and human and fruit fly and things like that, and they all have differences in terms of how they're reared and some of their developmental features, and it's always a struggle to apply techniques across these model organisms. And in this case, they're actually able to kind of do this across model organisms in a sense. They're able to kind of apply the same type of techniques and observe some of these things. So they also were able to do some interesting thing with the origins of blood. So actually to add stronger mechanical support for the embryos. So one of the problems, of course, is the mechanical integrity of the embryo. They use two different types of cell culture. So they used that cell culture and they added glucose to provide them with energy as they grew. So they were able to do certain things in the cell culture to make this, to provide stronger mechanical support as the embryos grew. When Tan and colleagues took a closer look at the embryo's mesoderm cells, they found that some had differentiated into heart muscle cells, and others had matured into cells found in the lining of blood and lymphatic vessels. So they're starting to get a circulatory system. They're starting to get some heart muscle, starting to get some vasculature that's growing, and they're getting perhaps some blood and other types of cells associated with that. The team also pinpointed cells that develop into connective tissues and one that form the foundation of the digestive system. So they're actually getting these precursors to certain parts of the different organs that are going to form. So not only are they getting like these precursors of the nervous system, but they're getting other precursors as well. And these are less farther along than the neural crest, but they're able to identify them. They also found signs that blood cells and their components were beginning to take shape in the yolk sac, which supplies the embryo with nutrients. These blood cells are almost impossible to obtain during human embryonic development. So this is something that they really, there's a lot of work to be done, but it's giving us sort of a good picture of early development and how these precursors sort of come into play or come into being. So I don't know if Morgan had anything more to say about that, or if you found that interesting. It looks like Jesse's back. SJ. Hello, Jesse. Okay. That's good. Yeah. So yeah, if you...
Sorry, Bradley. Just just getting kids ready for summer camp
yeah.
here. So, but it's super interesting and great that you covered all this. Yeah.
All right. Yeah. Thanks. Okay. I don't know if Susan had anything to say either, or.
I'm not that familiar with blood development, but yeah, it's an important part of development. I'd like to look at it further. I don't know. I need to finish projects so I can go off on my own some of the time.
Yeah. All right. Yeah. So let's see. Next thing I have is I'll go back to sharing my screen. And found an interesting thing on, let's see. There's a nice review of shear jamming that's come out. Oh, I don't think I have it here. Oh, maybe I put it here instead. Okay. So this is a nice review on shear jamming, which of course is part of the soft active materials that we talk about when we talk about the embryo. There are a lot of correspondence between what's going on in the embryo and what we see more generally with these soft materials. And so one of the things that we've talked about in the meetings and that come up in some of the migratory patterns of the embryo cell is shear jamming. And so this is a nice review. This is on the archive in the condensed matter soft materials section. It looks like it's really complete. It has a lot of, well, they gave a whole outline. I guess that's only 16 pages, but I mean, it's good. So this is the abstract here. Jamming is a ubiquitous phenomenon that appears in many soft matter systems, including granular materials, foams, colloidal suspensions, emulsions, polymers, and cells. So everything and then plus cells. When jamming occurs, the system undergoes a transition from flow-like to solid-like states. So we talked about these jamming phase transitions where you have flow through, say, like a pipe, and that flow can slow down with increased density. And at some point, there become so many particles in that pipe that slowed migration or that slowed movement stops. It just jams up. And there's usually some critical parameter that describes that stoppage. So this becomes a solid-like state, or it gets frozen into a state that's solid. And so you'll see this with some like water turning to ice, where you get water and the if it's flowing through a pipe, it thickens as it gets colder, and then it freezes solid. So this is a common phase transition in these kind of systems. Conventionally, the jamming transition occurs when the system reaches a threshold jamming density under isotropic compression. So this is under this kind of isotropic conditions, meaning that it's uniform across the material. And of course, it's not always the case. Sometimes you have anisotropies, so you're going to have differences in local jamming versus non-local jamming. We've talked about that actually with respect to some cellular systems where you just get these local jamming local jamming where you might get a cluster of things that become a solid and then the rest become kind of fluid-like. So that's kind of an interesting state as well, or a macro state, I guess. But recent studies reveal that jamming can also be induced by shear. So, of course, we've talked about shear and the role of shear in cellular mechanics. Shear jamming has attracted much interest in the context of non-equilibrium phase transitions, of which jamming phase transitions are one, mechanics and rheology of amorphous materials. Here we review the phenomenology of shear jamming and its related physics. We first describe basic observations obtained in experiments and simulations and results from theories. Shear jamming is demonstrated as a bridge that connects rheology of athermal soft spheres and thermal hard spheres. So these are, I guess, they're doing thermal characterization in terms of temperature dependence. Based on a generalized jamming phase diagram, a universal description is provided for shear jamming in a frictionless and frictional system. So they do this, they have friction and frictionless systems. We further review the isostasticity and criticality of the shear jamming transition and the elasticity of shear jam solids. So this is where we're looking at the shear jamming transition and some of its properties, and then the elasticity of some of these jam solids. So even though it's a solid, it still, has some soft matter properties that are interesting. They have a greater elasticity, say, than something that isn't. That maybe if you have like a brittle solid, for example, it's a little bit. There's a great elasticity there, even though it's a solid. The broader relevance of shear jamming is discussed, including its relation to other phenomena such as shear hardening, dilatancy, fragility, and discrete shear thickening. So this is kind of goes over different experiments, theories, simulations. So they have a series of simulations under different conditions. Isostasticity and critical scalings of the shear jamming transition, elasticity, and then related rheological phenomena. So this is these are the non-unique isotropic jamming densities. This is the jamming density here on this axis, and then they're simulating this in frictionless polydispersed thermal hard spheres in A. The jam states at the value J are compression quenched from equilibrium liquid states at EQ that are prepared using the swap algorithm. The density separates shear yielding and shear jamming behaviors in thermal hard sphere glasses. Then B is the dependence of this parameter on the interparticle friction coefficient mu sub 0 in simulated monodisperse athermal soft spheres. So this is where you have this parameter here. I think this is the isotropic jamming density on this axis. This is 1 over p hat. This is I guess this symbol, which is jamming density. And they show this for 3 different values, EQ, J, and C. And then this is where you turn this into an interparticle friction coefficient, or you look at the dependence of this on the interparticle friction coefficient. Oh, I'm sorry, this J is for this graph. So this axis is actually, this shows the dependence of this on, or this on this. So it's a different way of looking at it. This is the jamming plane here. So the J plane is formed by a large number of shear jamming lines. So this is for different parameter values. And that's then the experiments, they get into dry granular matter and dense suspensions. So this is shear jamming and granular experiments. This is a co-ed system used in the experiment of photoelastic discs where the state is unjammed. Then force network of a shear jammed state visualized by image processing techniques. So this is a force network here. We talked about networks last time with respect to embryo networks, but we also have these force networks that can form. Well, we've talked about some of these along the edges of cells, or along the edges, or kind of like at the some of the points within a mass of particles or something like that. So this is a force network of a shear jam state visualized by image processing techniques. And then this is the pressure strain curve here. And this is shear jamming accompanied with an abrupt onset of a finite pressure can be identified at a value of 0.2. So it's like right here where this shear jamming occurs, down here at this end of the function. So you have this phase transition where you have fluctuation around, this pressure, and then it falls off, and that's your phase transition. And so there's something that goes on in this network that changes when you get this shear jamming transition. And so it's kind of interesting how these...
Yeah, do they describe the image processing technique that they use to see that?
I imagine probably somewhere in the paper.
Okay, yeah.
Yeah.
If they don't, I feel like writing to them saying, how do you do this?
Shear jamming in simulations, so they were able to simulate this in different ways.
Oh, is that a stress strain curve?
Is it? I don't know.
Yeah, it is.
It is.
Yes. And it's
your
a
favorite
J-curve.
Okay, J-curve, great.
Yes.
So yeah, it's stress strain curves for...
I was looking for that foot. And yeah, so it's, yeah. Oh, well, yeah, it looks good.
Yeah. See, they got a stress strain J curve, and the other paper didn't. So that you were talking about earlier, yeah. So this is a unified strain density phase diagram of shear jamming, yielding, and what they call Gardner transitions in athermal soft spheres and thermal hard spheres. So the stress strain curves we saw in the last figure that I showed kind of translate into this phase diagram. So for these different values, you have, if you compare them across the range of values, you get these different regions that have like different properties. So for example, you have, so let's see, open cyan triangles represent the shear jamming line. So you have these triangles. This is the shear jamming line, obtained in athermal soft spheres. Filled blue triangles represent the shear jamming line, obtained in thermal hard spheres. So that's here, these blue triangles. Let's see if they have any other symbols here. Okay, but these show the different states that you get. So you get entropic flow. Here you get stable glass in this region. You get a jam solid here. You get mechanical flow here. And then there are these intermediary states that they don't, let's see, Mg is marginally stable glass. And then Sj is shear jammed. So these are different types of, like this is not a jam solid, this is shear jammed, this is a marginally stable glass. And then this is stable glass. So you can see there are these phase transitions, but sometimes you get these transitional zones in between them. And so you hit a limit, a physical limit, and you go over into a new state. So there are these different types of flow, entropic flow, mechanical flow. And so those are also, there's an entropic yielding line with the brown diamonds. That's entropic flow phase. And yeah, so that just basically characterizes these different, it's basically telling us that these phase transitions move us from one type of regime to another, and that there's some transitional regimes in there. This is shear jamming and two-step yielding, so this is kind of where they kind of get into the details of shear jamming, and they talk about that. There's this jamming phase diagram at zero temperature, so this shows like the jammed and unjammed components. There's isotropic jamming, which is IJ over here in this graph A, and then there's shear jamming on this side. So shear jamming is a different thing than isotropic jamming, and then there's the unjammed part of the graph here. And then you see this process of shear jamming, dilatancy, and hardening. So you get this, even within the jammed phase, you're getting these different processes moving up to an unjammed phase. So shear jamming is here, dilatancy is moving from isotropic to shear jamming, and hardening is moving within isotropic jamming. And so you can see that basically this isotropic jamming, I guess, would be something like a very stable solid. Shear jamming is something that's initiated through the shear process, or held together through the shear process, and then the unjammed state, of course, is what's not jammed, it's moving around. So nice physical characterization here. And so it's isostasticity is, as an isotropic jamming, the shear jamming transition always occurs under the isostatic condition. So this is what they're referring to when they refer to that. The removal of a single bond of an isostatic system could result in a floppy mode that extends in the entire system. Basically, it can introduce an instability. Thus, approaching the jamming transition, there is a diverging mechanical length, which resembles the diverging correlation length around a Stanhope thermal critical point. So there are these aspects of stability that are important here. And this is just where if you remove parts of the system, you can get different effects. So basically, as it becomes anisotropic, you get instabilities that either result in a phase transition or not. And so that's an interesting kind of point, because you usually think of, like, we don't really think of a lot of the sort of anisotropic aspects of this, where if you have variation in the system, what is the consequence of that? Well, especially over time, because these systems do evolve over time. So, and then they talk about approaching shear jamming from above. So they talk about summarizing the counterparts in isotropic jamming, which have been accurately measured in many studies. And they give a lot of math here that basically describes this. According to equation 10, which is this, the shear modulus G changes discontinuously at shear jamming, in sharp contrast to the case of isotropic jamming, so we talked about shear jamming and isotropic jamming being different, where G vanishes continuously at jamming. So this is G in equation 10, which actually is a shear modulus. So there's a discontinuous change of the shear modulus G at the shear jamming transition. And so these two things are equivalent. The macroscopic friction coefficient, which is mu, is also equivalent to that. And then it's also equivalent to P. So they're relating all that. Then they're talking about how G changes discontinuously at shear jamming, and that's different from isotropic jamming. In equation 6, which is up here, down here actually, G is related to how these two things here. The shear modulus G vanishes continuously at the jamming transition, so they talk about this set of relations. G vanishes continuously at jamming. This difference is explained by the elasticity theory. The last scaling in equation 11 is a consequence of the anisotropy of bond orientations denoted by a unit vector at distance r. So they're laying out the mathematical argument for this, and they're pointing to this difference between this sort of shear jamming and isotropic jamming, and linking that to elasticity theory. I think that's all, because they talk about related rheological phenomena, and then that's the end of the paper. And they have a lot of nice references here. This will be good for Susan, I think. 135 references on various things that might be useful to you, Susan. I'll send you this paper so that you can get
and could you send me the other one too?
Oh, yeah, I will.
I'm kind of interested in blood development.
Yeah.
Anyway, in contrast, say, to bone or something.
Oh, yeah.
Like, I always thought it's kind of interesting because bone is opposite of blood, but yet bone is what produces blood. Like,
Right.
Bone is the hardest material in the body, and basically blood is the softest or most flowing, of course.
Right.
And so I don't know. The contrast is neat, and then bone produces blood. So I don't know, just sort of
Yeah.
interesting. That's interesting. How does that work
Yeah.
in development?
Well, not only that, but you start out apparently with something that is like a set of stem cells that are differentiating into these components of the circulatory system, and then your bone comes into play, where it's the marrow is actually producing the blood or rejuvenating the blood, and that has to be kind of the switch over between just kind of producing this stuff through stem cells that are freely out there versus being in a place. I guess it's like we have stem cell crypts in the body where stem cells are produced, but I mean, it's kind of interesting how those things kind of get compartmentalized and they're in those places. And
Yeah, it's kind of an obvious place to look for stem cells, but that's
yeah,
in bone marrow.
yeah,
Anyway, it's interesting.
yeah,
Of course, our intestines have that stem cell niche in them too. So
yeah. Right. Well, I think that's all for today. Thanks for attending. I'll send out the papers. And, all right. Take care.
okay, well, thank you.
Thanks. Bye.
Thanks, Brenda.
Thanks.
Bye.
Bye.